Hello everyone and welcome back to our channel MooTechie. It's wonderful to have you with us for another session. Today we're shifting our focus from dairy to explore another fascinating and essential area of food microbiology, the microbiology of poultry and eggs. This is a topic that touches on some of the most widely consumed foods in the world, so let's dive right in. To start, let's look at some general context. Did you know that approximately 31% of all meat consumed worldwide is poultry? The only meat eaten more frequently is pork. The two principal types of poultry meat we consume are chicken and turkey, with turkey making up only about 14% of worldwide poultry meat production. It's fascinating to consider that the skin of live birds can naturally contain an average of 1,500 bacteria per square centimeter. These numbers likely reflect the natural flora of the skin plus other organisms that can be derived from the bird's feet, feathers, and feces. Now let's talk about what happens during processing. Contamination of the skin and the lining of the body cavity often occurs during washing, plucking, and evisceration. Most chickens today are processed on a fully automated conveyor line with vacuum evisceration. Bacterial numbers on the surface can vary considerably, but this variation is greater between different birds than between different areas on the same bird. At the end of processing, carcasses can have a population of mesophilic bacteria ranging from 10 to the power of 1 to 10 to the power of 4 colony-forming units per square centimeter. Every single step in processing influences the final level of spoilage bacteria, with some steps increasing the microbial load and others decreasing it. The types of organisms we find really depend on where we sample and the stage of processing. For example, the giblets, that's the gizzards, hearts, and livers, are processed separately, and the numbers and types of microbes there can differ from the main carcass. It's also important to know that finding Salmonella positive birds is common, with rates ranging from 0 to 50%. There is also a high incidence of Campylobacter jejuni in poultry plants and on the processed bird. The bacterial populations on finished carcasses are predominantly gram-negative and include groups like Acinetobacter and Moraxella, Enterobacteriaceae, Flavobacterium, and Pseudomonas. Now moving on to the crucial topic of spoilage. The bird's own enzymes contribute to the deterioration of the dressed carcass, but bacteria are the chief cause of spoilage. The intestines are the primary source of these organisms. Bacterial growth takes place on the surfaces, the skin, the body cavity lining, and any cut surfaces, and the decomposition products then slowly diffuse into the meat itself. The spoilage profile changes with temperature. Eviscerated poultry held at 10 degrees Celsius or below is spoiled mostly by Pseudomonas and to a lesser degree by yeasts like Torulopsis. Above 10 degrees Celsius, Micrococci usually predominate, along with growth of Alcaligenes and Flavobacterium. Over time, the surface of the meat will typically become slimy. Even small details matter. For instance, small amounts of iron, just 1 to 5 parts per million in the wash water, may favor bacterial growth and the production of a fluorescent pigment by Pseudomonads. Iced cut-up poultry often develops a slime accompanied by an odor described as tainted, acid, or sour, caused chiefly by Pseudomonas. Now let's crack into the microbiology of eggs, starting with contamination. Most freshly laid eggs are sterile inside, but the shells become contaminated very quickly by fecal matter from the hen, the cage or nest, wash water, handling, and packing materials. The total number of microorganisms per eggshell has been reported to range from 10 to the power of 2 to 10 to the power of 7, with a mean of about 10 to the power of 5. The types are very diverse. Importantly, Salmonella species may be on the shell or inside the egg as it's laid and can build up during processing. Next, we explore the spoilage of eggs. First, we have defects in the fresh egg itself, like cracks, leaks, loss of bloom, or stained spots. Inside, there can be meat spots, which are blood clots, general bloodiness, or translucent spots in the yolk. The changes during storage are divided into those due to non-microbial causes and those from microorganisms. Let's look at the changes not caused by microorganisms. First, untreated eggs lose moisture and weight during storage. This shrinkage is shown by the size of the air cell at the blunt end. A large cell indicates much shrinkage. Second, the physical state of the contents changes. The white becomes thinner and more watery as the egg ages and the yolk membrane weakens. When an old egg is broken onto a flat dish, the thin white is evident and the weak yolk flattens out or breaks. A fresh egg, by contrast, has a thick white and a yolk that stands up like a hemisphere. Also, during storage, the alkalinity of the white increases from a normal pH of about 7.6 to about 9.5. Now for the changes caused by microorganisms. To spoil an undamaged egg, organisms must first contaminate the shell, then penetrate the pores to the shell membranes. This usually requires a moist shell. They must then grow through the membranes to reach the white, and then manage to grow in the white, despite its hostile environment, to finally reach the nutrient-rich yolk. Generally, more spoilage is caused by bacteria than by molds, and these bacterial spoilage types are called rots. There are several types of rots. Green rots are caused chiefly by Pseudomonas fluorescens. The white turns a bright green in early stages, and the contents fluoresce under UV light. Odor is lacking or fruity. Colorless rots can be caused by Pseudomonas, Acinetobacter, or coliforms. The yolk usually disintegrates and can show a white incrustation. Odor varies from faint to offensive. Red rots are caused by species of Serratia and are relatively mild. Black rots make the eggs almost opaque when candled. The yolk becomes blackened and the contents turn muddy brown. The odor is putrid, with hydrogen sulfide, and gas may develop. Proteus species are the most common cause. Pink rots, caused by strains of Pseudomonas, are less common and feature a pinkish precipitate. Now let's look at spoilage of eggs by fungi. The spoilage goes through stages. Pin spot molding appears as small, compact colonies just inside the shell. Penicillium causes yellow, blue, or green spots. Cladosporium gives dark green or black spots, and Sporotrichum produces pink spots. Superficial fungal spoilage occurs in high humidity, first as a fuzz or whiskers on the shell. Many molds can cause this. The final stage is fungal rotting, where mold mycelium grows through the shell pores. This can cause jellying of the white and colored rots. The hyphae can weaken and rupture the yolk membrane, accelerating spoilage. Eggs can also develop off flavors. Mustiness can be caused by bacteria like Achromobacter purulens or by molds. A hay odor is caused by Enterobacter cloacae, and fishy flavors by some E. coli. Off flavors can even be absorbed from packing materials. Here is a helpful table linking bacteria to specific types of egg spoilage. Bacteria, type of rot, Proteus species, black rot, Aeromonas liquefaciens, black rot, Serratia marcescens, red rot, Pseudomonas maltophilia, green rot, Pseudomonas fluorescens, pink rot, Pseudomonas fluorescens, yellow rot, Enterobacter species, custard rot. Now how do we fight back? Let's discuss the preservation of eggs. The shell and its cuticle or bloom are the first line of defense, but they are porous. Membranes inside offer a temporary mechanical barrier, but this offers no protection against mold hyphae. A rapid temperature change can also help bacteria overcome these physical barriers. Fortunately, the egg white or albumin is an inadequate growth medium for many microbes. Its defensive characteristics are a high pH of 9 to 10 during storage, low levels of nitrogenous compounds, apoprotein Q, which ties up riboflavin, avidin, which ties up biotin, antiproteolytic factors, conalbumin, which chelates iron, making it unavailable, and lysozyme N, an enzyme that dissolves the cell walls of gram-positive bacteria. Now, let's explore the different ways of preservation. The first method is asepsis, which focuses on reducing initial contamination from feces and dirt. When eggs are broken for processing, care is taken to discard spoiled eggs and to sanitize equipment. The second method is removal of microorganisms. Washing can remove dirt and microbes but also removes the protective bloom and can encourage bacteria to be drawn into the egg pores. Using a disinfectant like a 1% hypochlorite solution in the wash water has been shown to reduce rotting. The third method is the use of heat. The heat coagulability of egg white limits the maximum heat treatment. For example, heating shell eggs in water at 60 degrees Celsius for 320 seconds can control rotting without causing coagulation. For liquid eggs, pasteurization temperatures are specified, 60 degrees Celsius for at least 3.5 minutes for whole egg liquid, and higher temperatures for yolk products. The fourth method is the use of low temperatures. This includes chilling for shell eggs and freezing for liquid egg meats. Shell eggs are selected by candling and should be cooled promptly after production. For long-term storage, temperatures of -1.7 to -0.5 degrees Celsius with 70 to 80% relative humidity are used. Oiling the shells is a common practice to slow down moisture loss and air penetration. For freezing, eggs are candled, washed, and broken on automated machines under strict sanitation. The liquid egg is filtered, standardized, and frozen. To prevent yolks from becoming gelatinous upon thawing, sugar, salt, or glycerol is added before freezing. Frozen eggs are stored at -17.8 to -20.5 degrees Celsius. Another key method is preservation by drying. Egg white often requires pre-treatment to retain its whipping properties. This includes fermenting to remove glucose brine, which prevents browning. Methods include bacterial fermentation, yeast fermentation, or using the enzyme glucose oxidase. Drying methods include spray drying, drum drying, and tunnel drying. The heat from drying reduces microbial counts but is not a reliable pasteurization step. The final moisture content greatly affects shelf life. Lower moisture, around 1%, is better. We also have the use of preservatives. This includes sealing the shell with wax or oil to create a barrier. For home storage, materials like salt, lime, or sodium silicate water glass solutions are used. Inhibitory chemicals like borates and benzoates have also been tried. Washing eggs with warm germicidal solutions is another application. Treating storage materials with mold-inhibiting chemicals and fumigating with gases like ethylene oxide have been used with some success. The use of controlled atmospheres is also important. Adding carbon dioxide to the storage atmosphere can delay spoilage, though high concentrations can affect flavor. Ozone has been used with mixed reports on its effectiveness, but it can have a deodorizing effect. Finally, we touch on the use of irradiation. Pathogens in liquid, frozen, and dried eggs can be inactivated by ionizing radiation. However, damage to the egg product has been observed at relatively low dosages, making commercial use seem unlikely. And that brings us to the end of our comprehensive look at the microbiology and preservation of poultry and eggs. It's a vast field where cleanliness, temperature control, and clever preservation techniques are all vital for safety and quality. Thank you so much for your time and curiosity today. We hope you found this session incredibly informative. Thank you. See you in the next class.